Hello, welcome back to Communicable, the podcast brought to you by CMI Communications, ESCMID's open access journal covering infectious diseases and clinical microbiology. My name is Emily McDonald. I'm a subspecialty physician in general internal medicine and scientist at the Research Institute of the McGill University Health Centre in Montreal, Canada, and an editor of CMI Communications. Candida auris is featured on today's podcast as the inaugural bug to be featured in our Nightmare series. I'm delighted to be co-hosting this episode with Narayanan, fellow editor at CMI Comms, an infectious disease pharmacist and clinical associate professor at Rutgers University in New Jersey.
Hey, Emily. I'm really excited for the guest that we have today. So our first guest is Professor Jeff Rybak. Jeff is a PharmD PhD and an assistant member in the Department of Pharmacy and Pharmaceutical Sciences at St. Jude Children's Research Hospital in Memphis, Tennessee. Dr. Rybak's primary research goal is to advance the treatment of patients with invasive fungal infections. Our second guest is Dr. Graham Snyder. Graham is the Senior Medical Director of Infectious Disease Quality and Outcomes and the Medical Director of Infection Prevention and Hospital Epidemiology at the University of Pittsburgh Medical Center in Pittsburgh, Pennsylvania. His scientific interests relate to infection prevention and control in multi-facility health systems and device-associated infections. Jeff and Graham, it's really an honor to have you on the show.
Great to be on the podcast. Thanks.
Thanks for having us.
It's time to open up the podcast with an icebreaker question. Actually, today's icebreaker had to be fungus themed. My question for the group today, what is your favorite culinary dish starring mushrooms? Nav, please go first.
I would be remiss if I didn't say it was something that my wife cooked. She cooks chopped mushrooms in curry type stir fry, which is really delicious. And then also my sister-in-law makes this stuffed mushrooms every Thanksgiving.
Jeff, you want to go ahead?
I'd have to say my answer is somewhat biased by wonderful experiences with colleagues at a meeting I recalled, a nice mushroom tamponade on some crusty bread with a selection of cheeses and cured meats would definitely be my go-to.
Amazing. Graham?
Yeah, I agree with Nav. You can't go wrong with stuffed mushrooms, however your house does that. But the one I'm curious to try is huitlacoche, or Mexican truffle. Apparently, it's a delicacy. I had to look up the fungus species. It's Ustalago matus. It's supposedly described as sweet and savory and earthy and woody and nutty. It just sounds delicious.
Okay, Graham, I'm really glad that you brought this up because I thought this controversy might emerge. Truffles, perhaps not technically a mushroom. I don't know if anybody came across this in their research. Mushrooms are said to grow above ground. Truffles are said to grow below ground. So they don't technically fall into the mushroom family, but I too chose a truffle dish and went with truffle risotto. We are starting off the podcast early with some intense controversy.
Emily, I don't know if you're allowed to host the fungal episode by picking a truffle, not being a true actual fungus. I'm excited to have you guys here. When we talk about emerging infections, the origin story is probably one of the most compelling things for me to try to understand. So I'd really like to start with maybe a little history lesson for our listeners. Could you tell us about this pathogen, maybe how it's discovered?
The initial report of the identification of Candida auris as a new species in the Candida genus honestly was somewhat mundane. It was an observation by Sato and Nakamura in Japan that a slightly different yeast organism that had been cultured from a 70-year-old woman's ear discharge, unclear whether it was truly infectious or just colonizing the ear canal at that point, had some unique growth characteristics and was genetically distinct from other known species of Candida. So they just made the effort to describe this new species among the hundreds of known species of the Candida genus that may have some clinical relevance down the road. At first, really the only features that were noted that were unique to this single isolate of Candida auris were its ability to grow at higher temperatures. In fact, the optimum growth temperatures were in the range of 37 to 40 degrees Celsius, which is a few degrees warmer than some of the Candida species most clinicians are more familiar with, such as Candida albicans. It grew only in a yeast form, even under conditions trying to promote pseudohyphal or hyphal growth. And it was able to grow and tolerate higher salt concentrations, initially tested at 10% sodium chloride, which again was somewhat unique. But really all that was notable from this initial report of what was a fully antifungal susceptible isolate of Candida auris. So this case occurred in 2008 and was initially reported in 2009. And over the next 3 to 5 years, we did see some additional reports of Candida auris being isolated and identified from clinical cultures, mostly in parts of Asia. There were a number of other cases in Korea that had been identified from ear canal samples, whether they be, again, truly infectious or just isolating from that reservoir or that niche. These are all genetically similar to that first isolate of Candida auris. Some of these, however, did exhibit elevated resistance to the triazole antifungals, especially fluconazole. But again, these were not truly known to be invasive infection isolates. And we're at this point up to about a dozen or so isolates of Candida auris that had been identified. But soon after this, we saw the first reports of invasive infection caused by Candida auris. And these were actually retrospective cases where, going back into some cultures, again from South Korea, they were able to identify isolates that had been previously misidentified or had not been speciated to a particular species of Candida. And this included 2 isolates from pediatric patients of approximately 1 year of age, and 1 isolate again from an elderly patient. All of these patients were in the hospital and had significant comorbidities, had recently been exposed to surgical procedures, had respiratory failure. None of them, however, were neutropenic. And these initial cases of Candida auris invasive infection were all candidemia, or a bloodstream infection caused by these Candida auris isolates. And unfortunately, 2 of these patients did succumb to their infections. However, again, only 1 of these 3 isolates, in fact, the isolate from the pediatric patient that survived their infection, was antifungal resistant with an elevated minimum inhibitory concentration for fluconazole. So this is really the first note that Candida auris may be a consequential pathogen for serious infections, and kind of the first call of attention to the organism. But from a global perspective, we really didn't see a large degree of attention being paid to Candida auris until a paper by Sean Lockhart and colleagues came out in Clinical Infectious Diseases in 2017, where we had the description of a simultaneous or near simultaneous emergence of outbreaks of clinical infection caused by antifungal resistant Candida auris isolates on 3 different continents within a span of 1 to 2 years. At this time, really, there had been some small reports of individual cases around the world, but many groups reaching out and from public health perspective, talking with each other, trying to identify whether or not this was a call for a great concern. Initially, and the CDC helped coordinate identification and handling of these infections, as well as a retrospective look at what the real threat here was. And what's really interesting is these 3 different near simultaneous outbreaks of Candida auris, which occurred in parts of Asia, South Africa, and South America, were later found to be caused by 3 genetically distinct subgroups, commonly referred to as clades of Candida auris. And these were actually distinct from those isolates that were initially identified primarily from patients' ear canals. These clades have since been renamed, numbered essentially, clades 1, 2, 3, and 4, and are associated with the initial geographic regions for which they were identified. But as time has progressed, we have seen this organism disseminate around the globe and have seen a number of outbreaks of infections on every continent outside of Antarctica, really highlighting the fact that while these isolates were initially identified in these different geographic regions, these clade designations are truly not an indicator of where you will find these clades, only where they were first identified. And during these outbreaks, we saw perhaps the most worrisome aspects of Candida auris, where isolates were spreading within medical institutions. We had a higher level of drug resistance to both the triazole antifungals and antiparsin B, and rates of mortality that were also worrisome. And around this time in 2017 is truly when Candida auris was recognized as a potential global threat to public health.
Since the pathogen's really only been known for about 15 years, is this a case of discovery bias, or did it really emerge simultaneously? Has it been around for longer, but once we found it, then we started to look for it? And now, are we missing cases? What does surveillance look like? I'm just wondering if you could both comment on that. Do we have a sense of mortality rates and if they're changing over the last decade? Maybe, Jeff, you want to go first?
I mean, there are many aspects of Candida auris that are troublesome and fascinating at the same time. And this mysterious of global emergence has been something that is asked about at scientific meetings repeatedly, trying to better understand why this organism seemed to come out of the blue. The initial cases of invasive infection that I mentioned, 3 patients that had cultures that were retrospectively identified as Candida auris, include one case that dated back to 1996. And that pediatric patient's case is, as far as I know at this time, the oldest confirmed case of Candida auris infection in a human patient. Folks have gone back through the repository of hundreds, if not thousands, of yeast isolates from around the world to try to see if perhaps there were Candida auris isolates that had just been misidentified or not speciated previously. And to date, that does not seem to be the case. Considering the proposed emergence in the range of the mid to late 90s to the mid 2000s, it's tough to see that there isn't potentially some connection with the large increase in the use of antifungals, both clinically and in other uses such as agricultural uses, as well as potentially changes in the environment in areas where Candida auris may be coming from naturally. It's tough to know for sure, but what we do know is that the isolates that have been disseminating around the world that belong to these majorly 4 distinct genetic subgroups are highly clonal in most cases, truly showing us the concept of One Health, how infections and outbreaks in North America can be caused by isolates that were traceable or at least genetically linkable to cases that are occurring all over the globe, and that we're really seeing a dissemination of this healthcare associated pathogen. And I honestly think much of our perception of Candida auris and its attributes, whether it be virulence, drug resistance, the ability to take hold in these medical facilities, is somewhat biased by the fact that the auris that we know is the one that we've kind of helped spread around the world and brought into these healthcare facilities. And we're really looking at isolates or lineages of isolates that have been successful in these healthcare settings and that have been able to survive drug treatment to the point where they can be repeatedly isolated. And we're somewhat predisposed to find a more resistant, more potentially problematic version of Candida auris. And as we continue to isolate more examples of Candida auris globally and find new genetic populations that are distinct from these highly successful clinical lineages, I think we're going to see a greater variety in the characteristics of Candida auris and might have a better understanding of what the natural version of this organism might have been before it faced exposure to antifungal drugs and perhaps other selective pressures.
We have a good number of case reports, case series, largely single center cohort studies. I agree. We're missing some high quality population based longitudinal surveys of prevalence, clinical presentation, and outcomes to give us the full sense. And
so from my reading, we're really only safe from Candida auris in Antarctica, and it's been found on every other continent. Can we discuss a little bit how Candida auris differs from other Candida? And from what I understand, it's usually transmitted as opposed to, say, emerging from the GI tract when someone gets sick or exposed to antibiotics. Do we know why it differs in that way?
Yeah, so any discussion of comparing and contrasting species of Candida probably is best to start by acknowledging the fact that the genus of Candida is kind of a catch-all for similar yeasts that grow in more or less white, typically smooth colonies on culture plates. Our nomenclature for this genus can be somewhat misleading at times because there's actually quite a bit of characteristic and genetic differentiation between different species of Candida that are really not that closely related. But one example compares the genetic similarity between Candida glabrata and Candida albicans, 2 more commonly known species of Candida, more commonly clinically encountered. And genetically speaking, it's said that they're about as related to each other as humans and fish. So there's quite a degree of genetic differentiation and characteristics within the Candida genus. And there's also perhaps 3 or 4 notable subgroups within this genus. Most of the Candida species that we encounter clinically, such as Candida albicans, tropicalis, and Paropsilosis, belong to a subgroup sometimes referred to as the Laudramyces clade. Clade just meaning they have a shared common ancestry that we can look at phylogenetically. Candida glabrata, on the other hand, Nachiomyces glabratus is its other name that is being touted now, is a much more distant group that is much closer related to Saccharomyces cerevisiae. Then we also have a group called Clavospora, and this is actually where we see some of the more drug-resistant and unique species within the Candida genus. This includes Candida lusitaniae, Candida hemolonii complex, and now Candida auris. And the interesting thing about this Clavospora clade of Candida is instead of being predominantly diploid like those of the Labromyces clade, they're typically haploid, and antifungal resistance is much more common among these species. The hemolonii complex are often found to have elevated MICs for amphotericin B, as is Candida lusitaniae and for Candida auris. As we look to why it may be something that we see more being transmitted as opposed to emerging from commensal flora, I think that is very much tied to what we were just speaking about, and the fact that Candida auris has really just recently emerged from an unknown reservoir or group of reservoirs in the environment around us. This is not a species that is part of the normal flora of us, any common vegetation that humans are coming in contact with, any animals that we know. Where we see Candida auris is really just going to be where it's been brought in contact with humans and where it's been able to spread and survive on these surfaces. And one characteristic that is definitely distinguishing for Candida auris is this ability to survive and spread on surfaces and take hold and persist for weeks, if not possibly months at a time. A lot of our impression of Candida auris as a clinical entity comes from these patients that have really first and foremost been put in contact with Candida auris. They've had an exposure where they even have an opportunity to become colonized or infected with the organism. And then more than likely, they also have to have one or more likely multiple risk factors where we've had antimicrobial exposure to disrupt their own normal flora and create some space for Candida auris to take hold. And in case of infections, probably also some conditions that alter their immune response to this new potential invader and give it an opportunity to get inside the human body.
I think one of the big gaps is about the interaction between Candida auris and our immune system, right? Jeff, you pointed out that this is not normal flora, this emerged from the environment. For me, at least, I think it's even more fascinating how this emerged as an environmental organism, but yet has characteristics to really evade our immune system, continue to inflict harm, despite not having much encounters as much as we know with the human immune system. Can we talk about that a little bit and understand really that interaction between this sort of new pathogen and our own immune system?
This is an area that we desperately need more research to better understand, but there are definitely some key characteristics of Candida auris that relate to its interaction with the immune system that seem to set it apart from other Candida species. The most prominent of which, the best described perhaps, is a distinct outer surface of the Candida auris cells that the immune system is going to be able to interact with. The Candida cell wall, the outermost layer outside of the cell membrane, has three major constituents or major layers to it, comprised of rigid chitin, a series of interlinked beta glucans, and then an outer layer of mannans, or these mannose containing carbohydrates that are linked to different proteins. And it's really this outermost layer that is potentially most exposed to our host innate immune cells. And one thing that has definitely been shown in multiple studies looking at Candida auris and its interactions with immune cells is that this outer layer of manna has a unique structure that has not been seen before in other species of Candida and perhaps other fungi in general, that gives these unique side chain features to these mannans, these branches that come off the side of the tree as opposed to extending it further up. And perhaps this altered structure that makes Candida auris more difficult to be recognized by our innate immune cells, such as neutrophils or macrophages. And there have been studies that compare the recognition and engulfment of Candida auris cells to that of, say, Candida albicans. And it certainly does appear that Candida auris is much less rapidly engaged with by our innate immune cells. However, there is also some signs that Candida auris, when it is engaged, induces a much stronger immune response with high levels of cytokine release. So there's kind of a push and a pull here where we have perhaps a lower level of detection. It also appears as though in vivo it might be able to replicate a little bit more quickly than, say, Candida albicans. but then also when it is recognized, a more robust and strong response, which may in fact lead to too strong of an inflammatory response. But that is very much something that's just been begun to be picked at, and not an area that we have a great deal of detail that we can look at right now.
We've heard a little bit about the differences between Candida auris and the other types of Candida. Does this mean that Candida auris is more predisposed to cause different types of infections? We heard the first case was found in the ear canal. What do we know about it clinically and how it differs and how it presents?
Yeah, acknowledging Jeff's comments about what's distinct biologically about it, broad brushstrokes, the clinical presentation is more or less what you'd expect for a Candida species. We focus a lot on serious infections, particularly bloodstream infections, intra-abdominal infections, wound infections. You may see urogenital infections, of course, otitis. And then there are case reports of different anatomical sites that wouldn't be inconsistent with what you'd expect for other Candida. So that's the early look at what the clinical presentations may be, also acknowledging that we don't have as many systematic evaluations of the clinical presentations. Mortality rates have been described in case series and some of these cohort studies to range from at least 25% to over 60%. There is probably some bias to focusing on bloodstream infections, and mortality from bloodstream infections due to a significant pathogen, they're not low. I mean, for example, for Staph aureus, we would expect at least 10% for all comers with a Staph aureus bloodstream infection. So this would be on par with a serious pathogen causing bloodstream infections. Asymptomatic carriage can be prevalent in high-risk populations, and asymptomatic carriage is an important part of case finding. It is more predominantly a skin commensal than a GI tract commensal. So when we screen for asymptomatic carriage and colonization, we typically focus on axilla and groin. Typical protocol is to swab both axilla and bilateral groin. For these reasons, and for reasons we've already talked about, it is well established to be transmitted in healthcare settings. So when talking about risk factors, I think the most important, significant first risk factor, particularly for a hospital epidemiologist like myself, is receiving care in a healthcare facility with Candida auris transmission. Now, this may vary from your global location of care to hyperlocal conditions, such as a specific long-term care facility. Regional prevalence may vary. In epidemiology, we partner closely with our public health colleagues to adjust our local risk assessment based on these factors. There's data suggesting collinearity between carriage of multidrug-resistant organisms, especially carbapenemase-expressing bacteria and Candida auris. So a multidrug-resistant organism and carbapenem-resistant organisms in particular are probably a risk factor because of commonalities of transmission. That gets to the second risk factor in healthcare-related risk factors. Having a central venous catheter, mechanical ventilation, parenteral nutrition, ICU admission, ICU length of stay. These are consistently described in the literature with odds ratios of 2x to maybe more than 10x. Or in case series, 25 to 50% or more of cases have these risk factors. That goes along with transmission in healthcare settings and risk of developing serious infection and not just asymptomatic carriage. Prior antimicrobial use, studies have shown that exposure to antifungal agents, both azoles and echinocandins, is a risk factor, but with a very wide range of reported risk or odds ratios. We've also seen antibacterial agents described. They're mostly described as a high frequency of prior exposure and not so much, say, robust case control studies or quasi-experimental studies exploring that risk factor. I think we have to learn from an antimicrobial stewardship standpoint about drugs that predispose to carriage and infection, as well as the advantage conferred from microbiome disruption, which some studies have started to detail the relationship between changes in the microbiome and Candida auris carriage. The last group of risk factors I talk about is a relationship between these healthcare factors, the antimicrobial related factors, and underlying health conditions that are associated with risk. One condition that comes up pretty commonly is chronic kidney disease, including chronic hemodialysis. Patients who receive chronic hemodialysis are at higher risk for multidrug-resistant bacteria too, and that has to do with healthcare exposure, interventions, factors like central venous catheters, comorbidities, etc. So these are deeply intertwined factors. But other ones that are described are recent surgery, including trauma, and that makes sense for surgical site and intra-abdominal infection risk, immunosuppression, diabetes mellitus, malignancy. Although these are less consistently described, alongside others such as respiratory and neurologic conditions. It's not clear that these factors are specific to the pathogenesis of Candida auris and or predictors of infection generally and collinear with other factors. But these are the types of risk factors that are showing up in studies across the board.
It sounds like any case of candidemia, then we need to think about an ophthalmology exam to check for endophthalmitis, remove all the catheters, potentially perform an echocardiogram, and then make sure that we have repeat cultures that are negative.
Absolutely. And you might also consider is there an intra-abdominal source and or complication would be another place to look. The other part of clinical management is making sure that your facility's team is aware, particularly infection prevention and the antimicrobial stewardship team. Healthcare facilities should have a response plan. Part of that is case finding, including asymptomatic screening and protocols for minimizing transmission. If you are concerned your patient has Candida auris, or if you've diagnosed an infection with Candida auris, or if you've identified it as asymptomatic carrier, it's not just the clinical management, but it's also the facilities risk management that's a part of it.
I think this is a point that's really well taken, is that it's not just at the human level, but in the environmental level, the sort of remolding that's happening out there has a lot of consequences. And so, Jeff, I think this will be a good place to dive into treatment. We could talk about your research, think about where we are in the world of being able to treat these infections and sort of what your lab is doing in this area.
So, unfortunately, discussions of treatment options for any invasive fungal infection tend to be relatively short because we have very limited options available. Really, as monotherapy for invasive infections, we're limited to 3 different major classes of antifungals. So we have the triazoles, such as fluconazole and posaconazole and the likes. We have the polyene amphotericin B and its various formulations. And then we have the echinocandins that are the only agents from this group that, instead of targeting something related to ergosterol biosynthesis, are acting upon cell wall biosynthesis, so mechanistically distinct as well. There is new agent that is clinically available, at least has been for other indications that are not for the treatment of invasive infections, but it is worth noting is it is a new class of antifungal that is clinically available. Ibrexafungerp, this has a mechanism of action that is similar to the echinocandins in targeting beta-glucan synthesis, but has a distinct structure and a distinct binding pocket or surface with the target beta-glucan synthase. However, this is currently only being used as an oral treatment for vulvovaginal candidiasis and cases where options are limited, and it's being used compassionately in serious infections. When we turn to the treatment of Candida auris, as the vast majority of our clinical isolates are observed to be resistant to fluconazole, and when I say resistant, and I'm going to personally be applying the tentative breakpoints that were set forth by the CDC a handful of years ago, based largely upon best available evidence, expert opinion, and susceptibility testing from around 300 isolates or so at that time, if I'm recalling correctly. It's important to distinguish these breakpoints from what we consider to be our typical or true clinical breakpoints, which are usually set forth with a large amount of clinical data to back up the in vitro susceptibility data to really give us a greater confidence that isolates that we're determining to be susceptible could in fact be effectively treated with these agents, as opposed to merely saying that isolates that are susceptible have lower MICs numerically when we test them in vitro. But at this time, this is really the best barometer that we have, and it was definitely a useful tool for the CDC to put out to give clinicians something to look at to better understand the organism that they're trying to treat. But again, unfortunately, over 90% of Candida auris clinical isolates tend to have very highly elevated MICs for fluconazole, our typical go-to anti-Candida triazole antifungal. The current recommendation from the CDC, because we don't really have any clinical data to back up any other practices, would be to use these fluconazole MICs as an indicator for the utility of the triazole antifungals to treat Candida auris in general, essentially meaning if it's resistant to fluconazole when applying these breakpoints, we should likely consider it to be resistant to the triazoles at this point in time as a class. Definitely in contrast to some other practices that we have with, say, the treatment of multidrug-resistant gram-negative bacteria, where we certainly can evaluate each individual drug from a class for its utility based on MIC. But again, here we do not have the clinical data to back up such a practice at this time. And because the resistance mechanisms that contribute to these elevated fluconazole MICs, at least among other Candida species, tend to impact all agents from the class, at least to some level, it's out of an abundance of caution that we kind of group these agents together with their susceptibility interpretation. Looking at the polyene amphotericin B, speaking very generally, we see about a 30 to 50% resistance rate to the polyene amphotericin B among clinical isolates of Candida auris. Some really great work that's being done by a number of groups right now to really show us that, at least for Candida auris, when we're testing susceptibility for amphotericin B, the method really matters. And the MIC that you get for any particular isolate can be dramatically different, whether you're using a broth microdilution assay, an automated susceptibility testing method, or something like a test strip diffusion assay. And the initial MIC breakpoints that the CDC determined, I believe, were all predominantly based off of a test strip diffusion assay at the time. And it's notable because if you take a pool of 100 or so Candida auris isolates and test their susceptibility for amphotericin B using various methods, you're not only going to get different values, but you're going to get different distributions of MIC values, suggesting that there's not really a good connection between the value that you get from one test or another, making it very difficult to know clinically whether you're dealing with an isolate that is amphotericin B resistant or not, when what might be the largest predictor is which tests you employed to determine what that MIC was. So I think this is an area where a lot more investigation is needed to really know whether or not there's still significant utility of the amphotericin B treatments for this particular pathogen. At this point, I still think this is relatively reserved as a second line or salvage type treatment. And truly, we're really leaning on or relying on the echinocandin as their primary treatment for Candida auris, where thankfully we still have greater than 95% susceptibility among clinical isolates as of today. Now, that being said, there are certainly a large number of reports of Candida auris isolates that either are found to be resistant to the echinocandins, largely due to mutations in that target gene encoding the beta-glucan synthase, much like what we see with other species of Candida, or having that resistance develop on therapy, which has again been reported multiple times. So it's always very important to look at your Candida auris patient and observe the progress of that treatment very closely, because the propensity of this organism to develop resistance on therapy may be different than what we know from other Candida species. When it comes to the work that my research group is conducting right now, we're trying to get some of the fundamentals and a better understanding of how our antifungal drugs work against Candida auris and which factors may be contributing to therapeutic failures. We do know that there are a number of well-defined resistance mechanisms that contribute to, say, triazole resistance, much like we see in other Candida species. And previously, we and our colleagues have shown that mutations in the target gene ERG11 and mutations in transcriptional regulators that control the expression of drug efflux pumps certainly contribute to triazole resistance in Candida auris and are found in a very large number of resistant Candida auris isolates. We're also now looking at potentially finding new sensitivities, new weak points that we could potentially therapeutically exploit for the treatment of Candida auris, and trying to better understand how this organism responds to the stresses that are caused by exposure to antifungals, and whether or not, if we were able to target some of these response pathways, we could get better efficacy out of our existing antifungal drugs. So this is definitely an area that we're focused on right now.
Hey, Jeff, do you see a near or distant clinical future for molecular methods to identify or predict an antimicrobial drug choice, like sequencing to identify the presence or absence of specific genes or nucleotide variants, particularly in the context of the limitations you outlined about susceptibility testing? Is this in the future?
At least for the case of the triazoles and the echinocandins, we can explain a large proportion of the observed antifungal resistance with some relatively small number of defined mechanisms that have known genotypes associated with them. So I do think that it's certainly something that is possible at this point. I would say either a sequencing method that can very rapidly acquire a genotype for a short list of genes that we know, or even small regions within these genes that we attribute to resistance, or whether or not we have molecular probes that can just identify these variants directly, which is definitely something that's being employed in other technologies at present. I think as we see additional populations of auris being reported clinically, where we're starting to see some different drug susceptibility patterns and perhaps even some different virulence patterns, those tests may be much, much more important. But unfortunately, it'll also be really important to make those tests available in the areas that really need them the most, which are often much more resource limited settings where advanced molecular tests are honestly not as practical. So I think that'll be up to us, again, under this concept of One Health, to know that helping detect Candida auris in these places where it's already a big problem and better treat it in those cases will help prevent it from spreading and help limit the problem before it is at everyone's doorstep.
Yeah, when you combine what you've taught us about variable frequencies of resistance that have been observed so far and some uncertainty there, plus limitations of conventional susceptibility testing, plus high mortality, at least what we've observed so far, including bloodstream infections, a really tidy solution is rapid molecular methods, either from growth or directly from blood cultures to guide effective therapy. And we've seen that elsewhere. It seems like it's a good opportunity to improve patient outcomes.
Yeah, I definitely agree. A key example of how simple that may be for the audiences of the initial echinocandin resistant isolates. And again, echinocandins are a frontline therapy and perhaps the most important class right now for the treatment of Candida auris infections. The vast majority, if not all, initial isolates, it's expanded since the initial reports, were known to be echinocandin resistant due to one of three different single mutations. So 3 different nucleotides changing, and all within a span of only a few nucleotides within an entire gene. So we would only need to know the sequence of a very, very small portion of this gene based on those initial isolates to be able to detect the kind of candida resistance. It has somewhat expanded since then. We're seeing more isolates with more unique mutations, but they do tend to be confined to these very specific hotspots that are actually quite small regions within a rather large gene.
I think if you're listening to the podcast and you've not yet treated Candida auris, your time will come probably because it's spreading. But nevertheless, the first take home is, echinocandins, either caspofungin or micafungin or nidulafungin, are recommended initial therapy, and they're clinically effective, and the likelihood of resistance is low. Amphotericin, liposomal amphotericin B particularly, is your next option. Should be considered if susceptibility testing suggests echinocandin resistance, or if you don't have response to therapy. And this is a clinical situation where, you need to make sure that there's response to therapy, such as clearance of candidemia. But the nuance here is, susceptibility testing plays a role because of limitations that Jeff's talked about with susceptibility testing. The second important take home is if you have diagnosed somebody with a Candida auris infection, you should have a conversation with your infectious diseases, your microbiology or antimicrobial stewardship experts about treatment selection, because it's a little complicated.
Yeah.
Graham, that's actually a really good point. When you've dealt with these situations, what has the interaction been like with the microbiology lab? I think thinking back to the reliability of the method, the reproducibility of the method, how it translates across different methods is all really important, and maybe something that's lost across clinicians.
Even before we saw a case of Candida auris, even before it was in our region, that's where the prep started. Can we identify Candida auris if it shows up? As the CDC gave us some great guidance on very early in the emergence of this pathogen, automated detection may go astray, may misidentify it. So the first conversation was with the microbiology lab. Say, hey, what tools are we using? What might misidentification be? And if we identify a pathogen that could be a misidentified Candida auris, what should our response be? Is it going to misidentify a very common pathogen and we don't panic yet? Or is the misidentification so reliably likely to be Candida auris that we should treat this as Candida auris? So that's a conversation that is the lab telling us about what the platforms are, what the misidentification might be, combined with infection prevention, antimicrobial stewardship, and our clinical partners and our administrative partners about, what is the appropriate response? Additionally, the conversation continues with, what happens if the lab finds probable or confirmed Candida auris? The standard would be just to report it out in the electronic health record, but this is an important pathogen. We want to do something more than that. You should already have a relationship in your organization with your microbiology team. If you don't, it's a wonderful time to walk over to the lab and say hi, because you want the lab to be a partner in a prompt response. Candida auris is not something we want to sit on either from a clinical standpoint, because we've talked about clinical complication of infection, or from an infection prevention standpoint. I think the key place to start with the role of microbiology, the conversation continues with what are the latest tools that we have to identify the organism, identify antifungal susceptibility.
And another aspect of Candida auris that is very much related to this interdisciplinary collaborative approach, knowing that because this organism is so good at colonizing surfaces and patients, and because many of the places where we see Candida auris being cultured from patients right now in the clinical setting, places like the urinary tract and the respiratory tract, in addition to more invasive, clearly infectious conditions, we have to really carefully differentiate colonization of spaces that we don't normally think of as being where Candida is truly causing an infection, and what may in fact be an infection of the urinary tract or the respiratory tract. There are a number of documented cases of Candida auris beginning in, say, a infection associated with an endotracheal tube, where they have clear signs of infection within the respiratory tract beyond just being able to culture the organism. That following treatment, the organism is then found within the urinary tract, where there may or may not be true signs of infection at the time. And as we have such limited number of treatments available for this organism, and many of these spaces are very difficult to effectively treat with our available antifungals. The echinocandins, for example, get very low concentrations within the urinary tract, or at least within the urine and the lower urinary tract, meaning that really reserving therapy for these cases where we are convinced that there is a true infection and not being necessarily startled by the initial isolation of auris, whether it be a colonizing case or an infection, and very carefully evaluating it, I think is going to be important moving forward to really preserve what limited antifungals we have left. And then again, communicating this plan with everyone so that if auris does pop up in another body site, which again may be colonization, there's not a snap reaction to initiate very broad spectrum antifungal therapy, which may limit our options down the road.
Yeah, it seems like people have to show a great deal of restraint, right? Because it would be very tempting to want to eradicate it once you found the organism in a person or in your institution.
We don't have treatment protocols for eradicating carriage like we do for MRSA. And we've already alluded to the potential for this organism to develop resistance. I usually teach that yeast in a respiratory urine specimen is very, very rarely anything more than an indicator of broad spectrum antibacterial use. But that also alludes to a little mini nightmare I've had, which is, could somebody have a Candida auris carriage that we don't speciate because it just gets reported as yeast? And so another conversation with the lab is, how do we not miss it? And there is no right answer to that, because many labs can't go speciating every yeast they find. And it may be situation dependent, like if you have other cases in your facility and it's going to be part of case finding. That's another example of an important multidisciplinary conversation that includes the lab and what are our lab methods? How do we modify them based on our risk assessment for Candida auris in this individual patient and in our facility?
Are we aware of any drug discovery ongoing for Candida auris, or any groups that are working on novel molecules to treat the disease?
So we do have some novel antifungals that are in the pipeline at various stages. I mentioned earlier ibrexafungerp, which is currently indicated for the treatment of vulvovaginal candidiasis, but at least at the time there was a plan to investigate it for systemic therapy for more serious infections as well. We also have notably some different azole-related compounds, such as the tetrazoles, so structurally related to, a little distinct from the trizoles, which may have different potency against Candida auris due to unique binding interfaces with their target, which may yield some additional utility from agents targeting sterile biosynthesis, targeting sterile demethylase itself. However, perhaps the most notable new drug in the pipeline, I think, from my perspective, would have to be fosmanogepix, an agent that has good potency against Candida auris in the initial in vitro test, and also a distinct target different from the agents that we already have, which may give us a new molecular space to probe with our antifungal therapy, where issues with cross-resistance might not be as problematic. This agent is something that targets GWT1 in Candida, which is a protein that is responsible for putting tails, GPI anchors, onto a number of different proteins that are necessary to be embedded or associated with the cell membrane. So essentially, the compound inhibits the organism's ability to get proteins where it needs them associated with the cell membrane and can cause cell death. There has been some early clinical use and some compassionate use for the treatment of various invasive fungal infections that does look promising. And it's definitely something to watch out for. I think an area where more investigation, more knowledge and experience may help us very quickly is looking back at our other triazole agents beyond fluconazole. I mentioned before we don't have the clinical data right now to back up the use of, say, isavuconazole or posaconazole for the treatment of a fluconazole-resistant Candida auris. But it is worth noting that when we have studied the resistance mechanisms present in Candida auris isolates that really drive those high fluconazole MICs, it's really just fluconazole and voriconazole that are very strongly affected by, say, the mutations in the ERG11 gene or by increased efflux pump expression. And these fluconazole and voriconazole MICs track very nicely together, suggesting that the cross-resistance there is probably going to be very problematic. However, when we look at the agents like isavuconazole and posaconazole, these ERG11 mutations, at least on their own, don't seem to dramatically affect susceptibility to these compounds. And some of the nastiest drug-resistant Candida auris isolates we have in our collection still have MICs for agents like posaconazole that are in the 0.25 milligram per liter range, which, especially with new formulations of the drug achieving much higher levels reliably in our patients, may mean that there's some utility left in this class of our most widely used antifungals that we could dip into in the future once we have that clinical data and experience to really support their use. I don't think we're there now, but I do see a potential in the agents that we have on the shelf right now that needs to be further explored.
Thanks, Jeff. Graham, you mentioned about colonization, about not over-treating. And what are those conversations like with frontline clinicians when they see Candida auris from a site that you might not believe is an infected site?
This is where the expertise of our antimicrobial stewardship colleagues comes to bear. And the themes are consistent with other conversations that we have with providers about judicious use of antimicrobials. It's a matter of educating people that Candida auris may cause colonization without infection. We have some limited data that patients can lose carriage over time, but we don't have any data that suggests the effectiveness of regimens to eradicate carriage. And I don't think there will be anytime soon, particularly given some of the things that Jeff was sharing with us about the biology of this organism and why it may be tenacious, but also the risk of developing resistance. That conversation usually means your patient is not the patient that we want to develop a pan-resistant Candida auris. And given the limited drug classes that we have, that may happen. Our antibiotic stewardship teams carry through themes and apply them to specific isolates and patients to help guide providers to judicious use of antimicrobials, and when they're indicated, selecting the right one.
How is the experience of dealing with things like carbapenem resistance and other types of bacterial resistance? How's prepared you from a hospital epidemiology standpoint in understanding infection prevention methods, contact tracing, and how you sort of deal with a single reported case of Candida auris in their hospital?
The program is similar, and you're highlighting carbapenem resistant organisms is apt because we start with the mechanism of transmission. And Candida auris is transmitted by contact, period. That's how it's transmitted. Now, contact may mean direct contact with contaminated hands. It may mean contact with contaminated surfaces, whether that be medical equipment or other surfaces in the environment. There is a little nuance. Candida auris is in general grouping of organisms that is sticky in the environment. Candida auris will readily contaminate the healthcare environment if a patient is asymptomatically or symptomatically a carrier of the organism. So we assume there is extensive environmental contamination with the organism in the care environment of a patient who is a carrier. So what do we do to respond with this type of threat? Well, we do contact tracing, which is case finding. This is being at risk of acquisition is a shared time and space with a person. Often it's the same unit. You might do for more extensive transmission that that has already occurred you might do for the entire facility. Case finding through identification of clinical cultures. This goes back to the topic of lab notification of a positive, lab notification of potential positive, lab identification of unspeciated yeast. And we also do asymptomatic screening. That's swabbing the axilla and groins of patients who are at risk of acquisition. When we have a patient who's a carrier, whether it's asymptomatic or symptomatic, we use contact precautions, gowns and gloves in their care environment and for direct patient care. Most facilities will also do some sort of enhanced observation of hand hygiene, personal protective equipment use, disinfection of reusable medical equipment to make sure that adherence is 100%. These are effective when done well. And then we mitigate transmission in the environment by using an approved agent. There's what we call the EPA List P. It is a list of products that are approved to be effective against Candida auris in cleaning of medical equipment and surfaces. These may be bleach, but they're not exclusively. In fact, different types of chemicals do just fine.
We talked a little bit about how the pathogen emerged. Is it at all possible that the warming of the planet had anything to do with the emergence of Candida auris?
This is definitely a hypothesis that has come up and been put forth by a couple notable people within the field of mycology. And I think initially it's impossible to exclude. It's definitely something that can be contributing. Considering the fact that Candida auris has the two unique features that came from that initial paper in 2009 that differentiated from other Candida species, its ability to persist at warmer temperatures, up to 40 degrees Celsius, 42 under more stress conditions, and the ability to survive in higher salinity than other Candida species, combined with the fact that of the few places where Candida auris has been found in the environment, we really have seen areas like estuaries along coastlines, areas in warmer climates around uninhabited bodies of water that make it seem as though Candida auris' place in the environment may be changing due to global warming. If you have coastline areas and warmer estuaries that are going to be some of the first impacted by changes in water level around the world, which is measurably increasing. Yet we're losing coastline, we're having increases in areas of this ocean water invading into land. We're potentially going to be changing the habitat of the organisms and microorganisms that live there. In addition, we have an increasing human population that's stretching out into areas that we're not previously living in. And we have this increasing use of antifungals all at the same time. But when you combine all these together, it may be no one factor, but I think all of them may be contributing to the emergence and potential spread of Candida auris.
This is not the Grimm's fairy tale where we had one zoonotic transmission at the human-wildlife interface, and we trace this all back to one patient zero. No. This is a scarier, in my opinion, Grimm's fairy tale, where the solution to it and preventing this type of event happening again is a concerted, multifaceted global response. This is a good One Health case study. That to me is a scarier Grimm's fairy tale in which we're all Hansels and Gretels and we're now all cookies.
I'll add that Candida auris is not the only new fungal pathogen that has emerged in the past handful of years. Just as that first case that was reported just identifying the species was not necessarily the cause of great fanfare, but more just a unique observation of a new fungi, we're seeing other organisms being identified now as well. We're certainly more likely to see this continue to occur if the environment in which these organisms are living continues to change. So Candida auris, unfortunately, may just be the canary in the coal mine, the first indication of this potential existing within fungi that warn us that we need to be on the lookout for the emergence of additional pathogens going forward.
On a more positive note, any thoughts on solutions to preventing the spread of Candida auris?
I think there's a few things, and I'd be very curious what Jeff thinks as well. I might want to know approaches to decolonization may be helpful to mitigate the spread. An expanded arsenal of antifungals as Jeff has previewed for us. So we've seen this with other organisms. There was a time when, maybe 3 decades ago, where MRSA was the boogeyman, and in part it was the boogeyman because we had few treatment options. And now MRSA, VRE, we have a bunch of options. So that aspect of the clinical threat is mitigated. And so the hope is with expanding antifungal options, the threat of Candida auris as a potentially untreatable pathogen diminishes over time. And then the third one is a clear understanding of the microbiome relationship with Candida auris, including carriage and development of infection, so that we can potentially modulate risk. I think we're probably at the precipice of a lot of innovation in the field of infectious diseases about the relationship between microbiome and pathogen, and changing the paradigm from find bug, kill bug, to create a less hospitable environment for the bug to be actually really cause harm. So those are the three things that I'm looking for in a hopeful future.
Yeah, I wholeheartedly agree that the better understanding of the implications of the microbiome and things that we can do to support good microbiota associated with ourselves and our patients is definitely an area of great untapped potential. 10 to 25% of patients known to be colonized with Candida auris go on to develop an invasive infection, really creating these conditions where we have the ability to maintain healthy microbiota, where we don't make room for Candida auris to take up hold. Our skin microflora is really going to help prevent a large number of patients being affected by this organism. One unique biological feature that we're beginning to understand better. I don't know that we have a good avenue right now towards new decontamination procedures, but where we are understanding more and more about Candida auris is why it's so good at colonizing a patient's skin, or how. And an interesting feature of the organism is, again, that high salt tolerance. And that, perhaps combined with some unique attributes of its cell surface, means that it's very good at adhering to skin, and that it's actually able to grow and produce better biofilm communities on skin than the isolates of Candida albicans that have been compared to it. So I think with this increased understanding, really putting an emphasis on decolonization and trying to contain Candida auris and prevent it from spreading is going to be our best avenue forward. And then obviously great new treatment options would be fantastic.
Jeff, that's well said. I'd love to hear both of your take-home messages for our listeners. We see a lot of nightmare organisms, right? And this is actually the kickoff, to our nightmare series. And so where does this stand in your sort of ranking of nightmare pathogens? Jeff, maybe we could just start with you, and then we'll give the last word to Graham on his thoughts.
Candida auris is this new and frankly still poorly understood fungal pathogen that has a unique proclivity to spread within healthcare institutions, much like other bacterial healthcare associated pathogens we're more familiar with, but in a way that we're not commonly associating with fungal pathogens. So we have to think differently about this organism from an infection control standpoint. Additionally, its high rate of antifungal resistance really challenges the clinical treatment of patients affected by Candida auris in a way where we are not uncommonly running into cases where we don't have a great treatment option available, either due to resistance or due to the type of infection that the patient has and our already limited antifungal armamentarium. I think going forward, our best opportunities right now, improving the state of Candida auris and from a global health perspective, is containment and identification, and really practicing high-level infection control practices, really identifying those patients affected early, communicating well not only within our own institution, but with other institutions as we care for these patients, and preventing that spread to hopefully slow its passage throughout healthcare settings around the world, so that eventually when it is on everyone's doorstep, we have these answers that we currently lack.
Yes, a similar sentiment as Jeff's. This is one chapter in the inexorable story of emerging pathogens. So the long view, this is another one, but it's important for our patients. We have a reasonable hope that we can minimize the extent of this threat, whether that be broad scale from the role of public health in tracking disease, providing guidelines for response, testing, treatment, to the things that each one of us can do. For each case, the roles of infection prevention control, antimicrobial stewardship, the microbiology lab, it has to be a team response. So we are on Candida auris. We know what to do, and it'll be a matter of executing and learning.
Thank you both so much, Jeff and Graham. This has been a really enlightening episode. As you all get to know me, I love puns, and so hopefully I'll be able to bring more to the podcast. The pun award for today, although you may have not known that you were competing, actually goes to Nav, who made reference to remolding the environment. Nav, I don't know if you caught yourself saying that, but it was excellent.
Come on,
Thanks,
Emily.
thanks, Emily.
Are you? Jeff and I had a couple that were not the yeast of the best.
Ooh, under the wire. Well played. The Nightmare series is going to continue with part 2 in early October. Thank you for listening to Communicable, the CMI Comms podcast. This episode was hosted by Nav, Narayanan, and myself, Emily McDonald, editors at CMI Comms, ESCMID's open access journal. It was edited by Katie Hostetler and peer-reviewed by Dr. Claudio Neidhofer of the University Hospital of Bonn, Germany. Theme music was composed and conducted by Joseph McDade. You can subscribe to Communicable on Spotify, Apple, or wherever you get your podcasts, or you can find it on ESCMID's website for the CMI Comms Journal. Thanks for listening and helping CMI Comms and ESCMID move the conversation in ID and clinical microbiology further along.